and how to produce microbial cellulose and lipase from Indonesian indigenous microbes, and also how to optimize cellulose and lipase from Aspergillus niger, immobilization cellulose for stabilization, and conclusion and acknowledgement. Okay, for introduction, Aspergillus niger, as we know that it's very potential fungi for producing the enzyme. It used for over 2,000 million years ago, and also microbial cellulose.
Excuse me, Aprida. Are you share your screen by yourself or?
I already give my presentation to
Presentation?
the.
Okay.
Okay, next,
Okay.
please. Okay, next, please. Okay, microbial cellulose. I will like to produce my own enzyme from Aspergillus niger to do the process, to do the production ethanol, and also lipase for producing the biodiesel. It's from indigenous Indonesian microbial. Okay, next. Okay, this is first of all, I do the screening Aspergillus niger and enzyme analysis. I'm using 4 kind of Aspergillus niger, and the most potential candidate is INACC F5 F538 code. Next. Okay, this is a material method. I'm using the Aspergillus niger as a isolate, and then I do the production lipase and cellulase, and I'm doing the powdering to produce a lipase powder and cellulose powder, and I do the optimization, temperature, pH, and concentrated using spray dryer methods. Next. Okay, this is the cellulose activity of Aspergillus niger. Uh, is a very got high uh, activity in 24 hours. It's 4.2 unit per milliliter. And then next. Okay, for the lipase activity, it's also in 24 hours optimization value. It's 3.7 unit per milli. Next. Okay, optimization pH and temperature of cellulase. The cellulase is got optimization in pH 7, and it's reached 1.72 unit per milli, and also for temperature, it's optimum. Not finished yet. Please go back to okay. For the cellulase, temperature of 50 degrees Celsius in 24 hours, it results 2.7 unit per milli and 3.11 14 unit per milli. Okay, next. For the lipase optimization pH and temperature, for pH, it's almost same with cellulase. It optimized in pH 7 and pH 8. It's more than 1.5 until 1.6 unit per milli. the range. And then lipase for optimum temperature is in 50 degrees Celsius. Is a range to 2.7 until 3.2 unit per milli. Okay, next. Uh, Actually, I'm doing two kind of process for powdering lipase and cellulase. I'm using the spray drying and also the freeze drying, and I do the comparisons between both of the methods. Okay, next. Okay, this is comparison immobilization content, cellulase and lipase. For the for the enzyme activity, the crude cellulase before using the spray dry, it's only 3.0 unit per milli, and after doing the purification and also the immobilization, it's increased until 19.6 unit per milli. And for the yield, it's reached 78.15%. And also the crude lipase as an activity before the immobilization, it's only 4.01, and after the immobilization process, it's increased until 24.1, and the yield goes to 82.37%. Okay. Okay, conclusion. Actually, Indonesia is a very giant biodiversity country, and he has a lot of microorganisms that very potential to produce enzyme, and it's our duty to do the exploration and not using commercial enzyme, buying from another country and very high and expensive value. We can do production with our own microorganism. Again, next, for the conclusion, second is okay. The second conclusion is my my I do the biorefinery and biotechnology to do the uh, new biofuel, and I will do the next experiment to use the cellulase for bioethanol research, and lipase for biodiesel research. And I will not using commercial enzyme or do buying from another country. Okay, next. The acknowledgements, I would like to LPDP scholarship grant for doctoral program and for financial support and work and scholarship. Next. This is the reference. Next. Next. Okay, thank you very much for your attention.
Okay, thank you so much, Ms. Aprida, for your really nice talk about these immobilizations enzyme. Okay, we go to the next presenter here, Ms. Karlaneta Kusumadwita, with the title Comparison of Calcium Carbonate Precipitation and Yeast Urea Culture Media on Hydrocarbonoclastic Bacteria Endospores Formations. Okay, Ms. Karlaneta, are you ready?
Yeah, I'm ready.
Okay.
Please commit to share my PPT. Okay.
Okay, the committee. How about the slide? Okay.
Okay, ya. Okay, Assalamualaikum warahmatullahi wabarakatuh. My name is Karlanita. Not yet. I would like to present about our research. The title is Comparison of Calcium Carbonate Precipitation and Use Urea Culture Media on Hydrocarbonoplastic Bacteria in Spore Formation. Okay, next. First, I would like to explain about the introduction. Concrete is the most commonly used in concrete material that has advantage high compressive strength, long durability, compatible reinforcement, and can be for as desired. But concrete has alkali and high temperature for the organisms. Several studies have been using hydrocarbonoplastic bacteria to increase the compressive strength in concrete, and it's called bioconcrete. Bioconcrete is applied for the fill the pores so that it strengthens the concrete structure and can readjust water permeability so that increase the compressive strength and tensile of concrete. Next, please. Hydrocarbonoplastic bacteria that can form the endospore are able to survive in the alkaline environment of concrete than a vegetative cell because bacteria in the spore can be stored for a long time and high resistant to the extreme environment of the concrete. Bioconcrete that contain hydrocarbonoplastic bacteria, if there is a crack, water and oxygen will have to enter and activated in the spore that could trigger formation of calcite through the enzymatic urease mechanisms. And calcite form will fill the crack so the crack will close back. Next. Okay, next. Media complexity affects the formation of bacteria in a spore, and one of the example is CCP media and U media. Several bacteria were isolated from the limestone mountain of East Java, which was Bacillus GA1, GB3, AK4, SU1, Bacillus GB2, and Sporosarsina GA4 that able to form endospores. In the study, the production and viability of endospore hydrocarbonoplastic bacteria cultures on CCP and U media will be compared. Next, please. Okay, next, the material and methods. First, for production endospore in CCP and U media, a colony in culture inoculates on CCP and U media and incubated for 24 hours. Then staining with malachite green and the endospore that can form calculated by hand telecounter. Second, Cellulose by shock treatment at 70 degrees Celsius for 5, 10, 15, and 20 minutes in water bath, and stored for 7 days and viability was observed. Okay, next. For the results of production of hydrocarbonoplastic bacteria in the spore, all isolates are able to form endospore in both media, in CCP and U media. In U media, the formation of endospore was higher than CCP, and Bacillus GB2 produced the highest number of endospore than other isolates. Next, please. For the results of endospore viability after 7 days storage, the longer the exposure time, the higher viability of endospores. With an exposure time of 20 minutes, is more endospore formed. Bacillus GB2 has the highest endospore viability compared to the other isolates. Next. Our study, we can conclude that first, all cultures are able to form endospore, and the production of endospore in new media is more than CCP media. Number two, Bacillus GP2 endospore was the highest than the other isolates. And the last is Bacillus GP2 endospores also has better viability than other isolates with soft treatment for up to 20 minutes. Okay. Next, please. Next, please. Okay, thanks. That's all my presentation, and thanks for paying attention.
Okay, thanks a lot, Miss Carlaneta, for your nice talk. And it's really fast. I think it's less than 10 minutes. Okay, it doesn't matter. We can continue for the next presenter is the title of the paper is Urais production by Basilisk using different source of urine substrate. So who's gonna be the presenter here? Miss Dia, yeah?
Rika.
Okay, Dia Rika, okay. Are you ready with your slides?
Yes, I'm ready.
Okay.
Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to introduce myself. My name is Diharika Rashida. In this occasion, I'm looking forward to talking with you about our research entitled Urease production by Basilisk sp. SK25 using different source of urine substrate. First, I would like to explain about growthing. Growthing is a method of filling growth material in pores and gaps between soil and particles. Growthing applications can be used to reduce the permeability of porous materials and change the structure of soil or sands. Growth material can be classified into suspension and chemical. However, the growthing method has a disadvantage because it can only be applied at the closest injection point. Therefore, we use biogrowthing as an alternative growth material. Biogrowthing method involves calcium carbonate precipitated from microbial metabolism as a growth material to strengthen the structure and reduce the permeability from the soil. In the general, the hydrolysis of urea by ureolytic bacteria is used as mechanism for the formation of calcium carbonate precipitates. Biogrowthing application can use bacterial cells with high urease concentration or directly by purified urease. Urease or urea amino hydrolase EC3515 is an enzyme that hydrolyzes urea into ammonia and carbonate. And this hydrolysis results in high carbonate concentration and alkaline pH condition that induce precipitations. And biogrowthing application using direct bacterial cells have some disadvantages. This is because bacterial injection is limited to fine-grained soil, and blockage easily occur when bacteria come into direct contact with solid grains in the soil. So, at present, the use of enzymes directly can be used as an alternative method. Therefore, in this study, the production of urease as a biogrowth material was carried out by Basilisk sp. SK25 using different urine waste substrates. So the aims of this study is to produce urease by Basilisk sp. SK25 by utilizing different urine substrates. In order to reach the purpose of this study, our methods begin with culturing Bacillus sp. SK25 and followed by producing and isolating urease. Then purification was carried out but by protein precipitation. After that, the pure urease characterizations was carried out by which included urease activity test, protein determination, and isoelectric point test. And the last method is by growing application. And this is the graph of bacterial growth profile of Bacillus sp. SK25 in urine medium was used as the growth profile of Bacillus sp. SK25 in urine medium was used as a reference for urease harvest time. And the isolation of urease can be carried out when the bacterial isolate has entered the stationary phase. Based on this graphic, urease can be isolated after 34 hours at 25 degrees Celsius and 18 hours after 50 degrees Celsius. And the production of urease was carried out through fermentation process. The result of fermentation by Bacillus sp. SK25 provides turbidity change in the production medium, as we can take a look from this picture. This picture B after 34 hours fermentation and at 25 degrees Celsius, and picture C after 18 hours fermentation at 50 degrees Celsius. And the urease activity after precipitation with ethanol is listed in this table. The highest activity of urease was obtained from the ruminant urine substrate at 25 degrees Celsius with an activity value of 102 unit per milliliters and the protein content of 0.019 milligram per milliliters. And the purification was continued by using ammonium sulfate. Based on this table, it is shown that the urease activity at the purification fraction of 60% to 75% was higher when compared to the crude urease activity before purification. And the isoelectric point of purified urease resulted in the deposition at pH 6, which characterized the coagulated urease protein characteristics. And this is the result of compressive strength test of the sand. And biographing application was performed using urease produced from purified from ruminant urine as a substrate. And the measurement of the repulsion power of the sand produced on average value of compressive strength in sand with urease treatment in graph A about 1.99 Newton, while in the sand without enzyme has a repulsion power of 0.76 Newton. So this is the conclusion of the study that urease production by Bacillus sp. SK25 with different urine waste substrates showed that the use of ruminant urine waste as a substrate gave the highest enzyme activity of 102 units per milliliters, and the resulting urea has a protein content of 0.019 milligram per milliliter with an isoelectric point at pH 6, and the resulting urease can be applied in biogrowing by giving an increase in compressive strength of 18%. And some advice for further research are there are some further there are need further optimization of the urine substrate is needed to determine and obtain more specific urea content so that it's easier to be used as an alternative substrate in urease isolation. And there needs to be a further study regarding the performance comparison of purified urease and urease directly produced by bacteria in biogrowing application. Thank you for your attention.
Okay, thank you, Miss Rika, for your nice presentation. Now we continue for the next presentation will be presented by Mr. Hendra Susilo with the title Diversity of the Cellulitic Bacteria in Rumen of Ruminants as a Probiotic.
Okay, I will present my paper. I will share. I will share the PPT. Okay. Okay. Good morning, everyone. Today I review journal. The the title is Diversity of Cellulitic Bacteria in the Rumen of Ruminants Potential as a Probiotics. Okay. Let's we begin of introduction. The main issue for developing countries, especially in Indonesia, is the agricultural waste. Uh, Agricultural waste in Indonesia has a low quality. Why? Because uh, the agricultural waste contains cellulose, hemicellulose, and lignins. All of them is a fiber that can difficult to degrade it. To avoid the accumulation of waste, the farmer make it to animal feed, but it cannot be directly used as a source of animal feed. So the livestock productivity is affected. To increasing the productivity, the farmer usually add the feed, the animal feed with a probiotic. Probiotic can keep the cattle health and the cattle more productive. Okay, let's we continue about the rumen. The rumen stomach is divided into four parts: rumen, reticulum, omasum, abomasum. Uh, but we focus in the rumen. Why? Because rumen has a lot of fiber. Uh, the fiber is in the rumen is such as cellulose, lignin, and the hemicellulose. And the rumen there is contain not only the fiber but also a microbe. So in this part in the rumen there is uh, occurs fermentation in the rumen. And if we want to degrade cellulose, we must add make microbe that can degrade cellulose. Uh, it will called cellulitic bacteria. And for the information, cellulose in the rumen is high concentrate. Okay, let's we continue about cellulitic bacteria. Cellulitic bacteria can degrade cellulose because the cellulitic bacteria can produce cellulase. The function of cellulase is to break the cellulose in the rumen into smaller fragment and monomer. We call it glucose. It can help the kettle metabolism. So we continue about the probiotic. Probiotics are microbes that intentionally aided dietary supplements and can benefit their hosts. Um, Giving of the live microbes in the sufficient quantities can affect the composition and ecosystem of the digestive microflora. Probiotic can reduce the population of the bacteria, pathogen bacteria. Probiotic can also act as alternative ingredients for growth enhancer. So for the information, probiotic can increase the uh, hormone called thyroid. Thyroid can help regulate the body metabolic rate. Good metabolic can help, uh, it can also increase the production of milk and meat. This is the characteristic of probiotic. Uh, the microbial requirement is classified into probiotic is one, resistant to acids and bile salt. Two, adhere to intestinal mucus. Three, produce antimicrobial substances. We call it bacteriocin. Four, high viability and stability. Five, safe for the digestive tract of animal. This is the role of probiotics. We can see this picture. It can help metabolic effect because probiotic can produce bacteriocin, decrease pH. Uh, they can also colonization resistance because they can kill the pathogen bacteria. And probiotic can also give the can stimulate immune system. This is this picture. I am focused in the bacteriocin activity. Bacteriocin can defeat the pathogen because bacteriocin can create formation the pores of the inner membrane. It can also damage the nucleic acid pathogen bacteria, and it can damage the precursor of peptidoglycan of bacteria, pathogen bacteria. Okay, next to rumen. Rumen is a complex ecosystem where the nutrient inhabited by anaerobe microorganism. The interaction between microorganism and host animal produce symbiotic relationship that allows ruminants to digest a diet rich in the fiber and low in protein. This is a structure of cellulose. The cellulose chains consist of glucose, glucose, one glucose and one another glucose is bounded by beta 1.4 glucoside bonds. Then this is cellulase. Cellulase is consists of three enzyme complex, namely endoglucanase, cellopiohydrolase, and beta glucosidase. Okay, we'll continue about the cellulosic activity. The cellulosic activity can be measured in two ways: qualitative analysis and quantitative analysis. Qualitative analysis by examine the diameter of clear zone, and this is quantitative examination by adding DNS reagent. This is the table of cellulose enzyme activity, cellulomonas. We can see in here all of the genera cellulomonas in here. It can produce a high activity enzyme. This is a mechanism of cellulose complex in the three steps. Step one, cellulose is hydrolyzed by endoglucanase by form the fragment cellulose, small fragment cellulose. Step two, cellulose is further hydrolyzed by exoglucanase it can release small oligosaccharides and cellulose. And step 3, beta-glucosidase can hydrolyze cellulose into glucose monomer. This is identification of cellulosic bacteria in bovine rumen. I get the species. I get the species. I get 50 species in NCBI from the accession code. I get the accession code from the journal last 10 years. This is about all of the species in the dominant family is Leptospiraceae, Ruminococcaceae, and etc. Prevotelaceae. This is bovine rumen. Not only bovine rumen, we can see the cellulosic bacteria. It can found in the sheep rumen and mouse rumen. So the ruminant wild and domestic ruminant, we can see the cellulosic bacteria. Okay, this is a simulation of phylogenetic tree. I use the Mega 5. In the scale in here, it represented about the similarity index. The similarity index, I give example for this. In the large similarity index, it showed that Acinetobacter and Pseudomonas is related. In the phylogenetic tree, we can see this is the family of the diversity family of the bacteria rumen. We can found Prevotelaceae, Lactospiraceae, Eubacteriaceae, Spirosetae, Spirosetaceae, Closidiaceae, Chrominococcaceae, Streptococcaceae, and Basilaceae. This is the tree analysis phylogenetic tree. The percentage value of the similarity between 50% in the we found it in the Clostridium autotolerant and Pseudovibrio ruminis, and the highest 100%, namely in the pair of Prevotella ruminicola, Prevotella brevis, Butyrovibrio proteoclasticus, Butyrovibrio hungate, and etc. In the genus of Cellulomonas and Prevotelaceae were reported to have the highest cellulosic activity. This is a potential probiotic based on cellulosic activity. So the potential of probiotic in ruminants is related to the function of microorganism in the rumen by changing the low quality. The mechanism is two. One is improve fiber digestibility, and two, accelerate VFA volatile fatty acid. With the addition of probiotic, feed containing high crude fiber will be digested by a cellulosic probiotic group because it is able to produce cellulose enzyme. So animal can utilize energy source more quickly. Increasing VFA can increase lipid concentration in milk production. Okay, this is a conclusion. We know that cellulosic bacteria, the family is Prevotelaceae, Lactospiraceae, Eubacteriaceae, Spirosetaceae, Eubacteriaceae, and Ruminococcaceae. Cellulomonas and Prevotella has a highest cellulosic activity, and the analysis of phylogenetic tree in based on 50% until 100%. The addition of cellulosic bacteria probiotic together with feed will improve fiber digestibility, stabilize pH, accelerate VFA change, increase feed consumption. That's all about my presentation. Thank you for your attention.
Okay, thanks a lot, Mr. Hendra Susilo. I think that was really awesome. So we can eat our informations about, it seemed like a review, right? So yeah. about the diversity of the bacteria in a rumen. Okay, thanks a lot. Now we move to the last presentations in these sessions. The paper with the title Fungal Applications in Bioflotation of Sinking Fish Feed, Possible Roles of Surface Mycelium by Mr. Chatur Sri Herwanto. Okay, Mr. Chatur, time's yours.
Okay, thank you very much. Can you please, can you see my presentation?
Yeah.
My slides?
Yes.
Okay, good. Assalamualaikum warahmatullahi wabarakatuh. So good afternoon, everybody. Thank you for the organizer for having me here to present my research, my group's research titled Fungal Application in Bioflotation of Sinking Fish Feed, a Possible Role of Surface Mycelium. So as we know, tempeh is the fermentation product of soybeans using tempeh starter, or in the laboratorium context, we normally use the Rhizopus, the fungi from genus Rhizopus. Due to the scientifically proven nutrition content of this tempeh, now tempeh has been has been consumed in worldwide, and also has been used not only for fermenting soybeans, but to ferment any beans which are locally available. It's not limited to soybeans. More importantly is, in the last few decades, the Rhizopus fermentation has not only been limited to be studied for human consumption or human nutrition, but also has been extended in the study of its usefulness in the nutrition for the animals, like cattle, like birds, as well as fish. Particularly for the fish, Rhizopus has been researched, and numerous studies has produced results, which is Rhizopus contain high amount of proteins. Some say it is about 40 to 62% of protein, and then it contains also essential amino acids, which is comparable to the fish meal, which is normally used in the fish feed. It has high protein digestibility, up to 80%, and what importantly, Rhizopus is easily grown, and it can use various substrates, especially waste and also agricultural byproducts, which is very cheap. So now I'm showing you at least two types of feeds. On the left is the commercially floating fish feed. This floating fish feed is usually produced using a very high-tech machine called extrusion machine. And this machine is not only very expensive, but it also needs a high skill in order to operate it. It needs a high energy input, and it needs a constancy in the formulation of the feed. And this feed is very expensive. So that's why for the moderate and small scale fish farmer, they usually don't use 100% of this feed, because if they use it 100% from the beginning until the harvest time, then they will get very low profit. So normally toward the end of the harvest time, so maybe 2 or 3 weeks or 1 week before the harvest time, they switch into the low price feed, or sometimes they produce their own feeds, which is on the right. But this feed is very low quality, and it easily degrades. It breaks down in water, and it sinks in water, because they do not have machinery, extrusion machine, in order to produce floating feeds like the commercial floating feed. So this is problem, because if they use this easily degrading and sinking feed, then the feed not only, most of the feed is not only being wasted and go down into the bottom of the pond or the water and pollute it, but it also reduce the effectivity or the efficiency of the feed use during the fish farming. So what we are doing, what we have been doing so far is we are trying to find an alternative method of making the sinking feed to float without any machines, which is very cheap and very easy and hopefully applicable to the farmer in the field. What we have been doing is we use fungi, which is tempeh fungi, in order to produce the buoyancy or the floating property on the feed. So this is very simple, and but there is some question to be answered in this method. What actually causing the feed, the fermented feed, to float, and how actually this happen? So these floatings happen. So the objective of this study is to demonstrate the possible role of surface mycelium in the fungal-induced bioflotation of sinking aquafeed. Okay, so the steps that we use in our research is, the first is solid fermentation. So we do the fermentation like what the tempeh, the people making tempeh, so it's solid state fermentation. And then after that, we do some treatments to the fermented product. And then after that, we did physicochemical analysis. So this is the first step is solid fermentation. So we use commercial sinking feed. We didn't formulate our own feed because our concern is not the nutrition but the physical properties of the fish feed. So this is, we got a sinking feed which is normally for shrimps. And what we did is we pulverize using blender, and then we mix with the water to have about 35 hydration or moisture, and then we mix with tempeh starter or tempeh inoculum, which we purchased locally near our lab. And then after we mix homogeneously, then we transfer it into petri dish, inside of which we have like a frame, okay, like rectangular shape frames, and we incubate for about 28 to 33 degrees centigrade for 48 hours. So this, after the fermentation, we get the fermented sinking feed like the one in the picture. So we see the surface of the petri dish was covered by cotton-like structure. So it indicated that the fungi, the normally rhizophus, grew very well. And then we did 3 different treatments. Okay, the first treatment is actually we didn't do fermentation. We just directly do the oven drying. So the sinking feed without any fermentation. And the second treatment is after the fermentation was resulted, then we peel. So we peel the surface of the surface mycelium. We peel like we are peeling what you call it, apple. So we remove the skin completely. And the third treatment is we didn't do any peeling, so we just do the oven drying directly. And then after oven drying, we did 7 analyses, which consisting of 5 physical analysis, and the last 2 is chemical analysis. So the 5 physical analysis include the density, floatability, water stability, hydrophobicity, and water absorption rate analysis. And the chemical test includes soluble protein, carbohydrate. So this is the macroscopical appearance, so before and after fermentation. And this is after 3 different treatments. So on the left is the feed without any fermentation, so just oven drying. And as you can see here, the particles making up the feed broke down easily. And then in the middle and on the right, as you can see, the structure is preserved. Okay, the cubicle or the block structure is preserved, indicating that the mycelium of the tempeh mold function like what do you call it, joining the particle of the feed to make a single structure and not easily broke down. And on the in the middle one is the one with mycelium surface intact, so without any peeling. And on the right is after the peeling, so the mycelium, the surface mycelium is completely removed. And then we did the first test, which is on in using water. As you can see here, in the first minute of the test, the unfermented one completely sank at the bottom and disintegrated, so break down easily. But the unpeeled fermented feed and peeled fermented feed, it didn't disintegrate. But the one without surface mycelium, then it sank. Whereas the one with intact mycelium, surface mycelium, it float better. It can float better than the others. So this is the graph of the floatability, which is to measure how good the feed can float on water. So as you can see, the one without fermentation or unfermented feed control, it sink right at the beginning of the floatability test. And then in the middle is unpeeled fermented feed. As you can see, it float until the end of the floatability test, which is 2 hours. Although the floatability was decreasing gradually, but it's still better than the one at the right, which is peeled fermented feed, which is the fermented feed with the surface mycelium removed completely. As you can see, although at the beginning it floated all of them 100%, but within the first 2 or 3 minutes, it completely sank at the bottom. So then we measure the hydrophobicity of the surface of these 3 feeds. As you can see, unfermented feed, when we tested using a droplet, the water droplet was easily quickly absorbed by the surface, and it took only 0.63 seconds to absorb completely the droplet, and the hydrophobicity or contact angle is less than 90%. So sorry, 90 degree. So it means it is not hydrophobic; it is hydrophilic. But in the case in the middle and on the right, which is fermented feed, as you can see here, when the surface of the mycelium was still intact, so the water droplet was not easily absorbed. So it took about 20 seconds to absorb, compared to the one on the right, which is without surface mycelium. It was more quicker than to absorb the water. And as you can see here, the one with mycelium intact has the only hydrophobic contact angle, which is more than 90 degree.
Okay, Mr. Chatur, walk a little bit faster.
Okay, I will be finished at 2 more minutes. Okay, so the possible explanation is maybe the mycelium contain this chitin and chitosan, as normally the cell wall of the fungi do have. So this chitin and chitosan have a hydrophobic nature, as well as the protein called hydrophobin. So hydrophobin is the proteins normally found on the filamentous fungi, which is also hydrophobic. And the last slide that I want to show is from the result and discussion is, although it did successfully make the feed float, but unfortunately there's a drawback here. So the water, as you can see, the water became cloudy because of this fermented feed. So it means there were some nutrients which leach into the water, and we measured this leaching leaching nutrients or compounds. It could be because of the protein and carbohydrate. As you can see here, there is an increase in the soluble protein and the sugar in the water in the fermented feed, both unpeeled and peeled, compared to the unfermented control. So my conclusion is that The surface mycelium did show hydrophobic properties that slowed down water infiltration to the feed matrix. It maintained the density below that of water and thus prolonging the flotation duration in water. But further studies is needed to find a method to prevent nutrient leaching from the fermented feed. So thank you for the attention, and this is also the contributors of this research. So thank you.
Thanks a lot, Mr. Chatur, for your really clear explanations about the potential of the fungal. Okay, also all of the presenters have been present their paper. So now we continue for the Q&A sessions. Is there any question here? And you can maybe you can raise your hand and then you can ask the questions. Okay, not yet. Okay, not yet. Okay, well, we maybe I have a questions. For the first, I want to ask to Mr. Chatur about is there possibility to use any other kind of fungal? I know that especially for individual people, they have a lot of fermented foods. So can you tell us we can use other fungal?
Yes, it is possible. Yes, the reason why we use rhizopus is uh, because rhizopus, the research on rhizopus for the animal or fish feed is plenty numerous. So, and they have shown actually the result of using rhizopus to substitute fish meal in the fish culture. So yeah, so the reason why we use rhizopus because rhizopus has been shown to be applicable in animal or fish diet. So of course you can try other edible fungi, not only rhizopus. So as long as this fungi can actually colonize or can grow on this fish feed as well, just like rhizopus, then yes, it is possible to make the fish feed floating on water. Yes, thank you.
Okay, so the key point here is maybe we can choose any other edible fungal. Is that right?
Yes, the main thing is it is edible and it is grass. Okay, it's recognized generally as safe. So because it's on for consumption of the animals and the animal will be consumed by human. So you have to make sure at the beginning that this fungi is safe for the animals and also for human.
Okay, thank you so much for your explanations. Okay. Okay, is there not any question? I think the time's up. So, but before we leaving the sessions, what you mind to we take a picture first, and then after that we can close the sessions. Okay, maybe Helen or Edo can help us.
Okay, I will write the photo session today. So all the participants of the Zoom, I would like to ask you to turn on the camera, please, because we are going to enter the photo session. Okay, there's only one page here, so there's some half of the participants who haven't turned on the video yet. Please, okay. Okay, that's for now. So I will count 1, 2, 3. Okay, that's it. Thank you so much.
Okay, thank you, Helen. Okay, so I think that's all for the sessions. Thanks a lot for your really nice participations, and thank you so much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.